moving one of the stations if you're practical is to do a pulse work test. Because just if you can ask anybody who works in the lab, they do those all the time, so you know how to do that. So who in here can do a PowerPoint presentation today? How many are left?
Just,
we got
you raise your hand more.
So we got this week and next week. All right, that'll give us, well, I'd like to have them done at the end of next week, the PowerPoint presentation. We're on chapter 10.
Semen.
I did look at the PowerPoint for chapter 10 in D2L. I really liked it. That's a very good study guide for your test. Okay, so even though we're going to go through the book, I'm going to have you do the same thing. Underline what's important out of this chapter. If you want a good study guide where you can see some of these pictures that, you know, up close, that's a good study guide. All right, one of your first objectives has to do with the structures involved in semen production and composition. So if you will, turn over to page 204 with me in your textbook. We're going to have to talk about a subject today called andrology, and that is basically the study of the male reproductive system. Okay? Depending on where you go to work at, you may be like myself, and I have never had to do a semen analysis. Jason, on the other hand, has, because the hospital he worked at, they did post vasectomy semen analysis. Where I worked at, we did not do it in the main lab. It was an outpatient procedure. So, you know, again, I always let you guys know I never actually did this, right? So I'm teaching it from the book. But it is something that is done, and it is an increasing field because they have andrology labs now, because there's so much technology that is advancing about infertility and conception in vitro, that this could be a booming field. All right, I just want to tell you in this chapter that semen analysis is one of the high complexity tests that's done in the lab. You guys know what I mean by high complexity. High complexity means that you cannot do a semen analysis because there's so much interpretation that has to be done. It's so manual as far as loading hemocytometers and things like that, that this is considered one of the more advanced type of lab techniques that there are. All right, so again, when I mention high complexity testing, there's wave testing. Wave is like those little kits you guys do in there. That's real simple. Anybody can do those, so that's considered wave testing. But once you get to the moderate complexity or high complexity, you are not allowed to do those tests unless you have graduated from an accredited program like this one. And in most states, you have to be licensed or certified before you're able to do this. Okay. All right. So what makes up a complete semen specimen and the anatomy that's involved with it? We're going to talk about the testes. We're going to talk about something called the epididymis, the seminal vesicles. We're going to talk about the prostate gland, and even there's something called the bulbourethral glands in the reproductive tract of a male. All of these little organs, I call them, they have to do with development of the entire semen specimen. Okay, because we in the lab, we are going to be testing somebody, of course, by masturbation has to submit a sample. Okay, they usually do that either at home or they have special rooms for that. But the reason that I think it's important that you know the anatomy of this is because it has to be a complete specimen. For example, and I'm not trying to get gross on you guys, but if they don't collect the first part of the specimen, let's just say for whatever reason they don't do it right, and the first part of the specimen doesn't get collected, there's a good chance that you may not actually see sperm in that specimen, because the very first portion of that contains the sperm. Okay, so this is a real good objective, and there is a test question on it, and it's worth a lot of points. Okay, so we're going to go through this, kind of talk about how the sperm develop, and what all goes into the entire specimen. All right, so we're going to start off with the testes. All right, the testes are the seminiferous tubules. This is where the sperm actually develop. Okay, by develop I mean this is where they originate from. Okay, they're called like germ cells when they start off. So under the physiology on page 204, the left-hand column, it says semen is composed of 4 fractions that are contributed by the testes, the epididymis, the seminal vesicles, the prostate gland, and the bulbourethral glands. So the very end of that column, there's 2 words. It says germ cells for the production of spermatozoa are located in the epithelial cells of the seminiferous tubules. Okay, they're talking about in the testes. This is where it originates from. This is going to be about 5% of the fluid, right? And we're talking about sperm. Now, if you'll drop down a few lines, it talks about the epididymis. It says the epididymis is where the sperm mature and they actually develop the flagella. So everybody look at that picture at the bottom of page 204. I just kind of want you to understand what we're talking about. You obviously see where the testes are located, right? See that little gland located just above the testes? That's the epididymis. Everybody see that in figure 10 tac 1, the picture up above? All right, skip down to the next paragraph, and it says the ejaculatory ducts receive both the sperm from the seminal vesicles, produce most of the fluid present in semen. This is going to be about 60% is the seminal vesicles. It goes to say when they're talking about fluid from the seminal vesicles, it says, I'm in the last 2 lines in the right-hand column. It says spermatozoa metabolize fructose for the energy needed for the flagella to propel them through the female reproductive tract. In the absence of fructose, sperm will not display motility in the semen analysis. So the seminal vesicle is what's responsible for that fluid, okay, that basically causes the sperm to metabolize or utilize fructose so they can move, right? Yep.
And this fructose is sourced from diet?
What's that?
The fructose, it's sourced from diet?
Yep. Okay, let's talk about the prostate gland. I'm on page 205, go to the first paragraph and drop down about 4 lines. It says approximately 20% to 30% of the semen volume is acidic fluid produced by the prostate gland. This milky fluid contains a high concentration of acid phosphatase. You guys will remember that's one of the enzymes that come from the prostate. Remember, prostate acidic phosphatase. All right. It also contains citric acid and zinc. It contains enzymes responsible for both the coagulation of a semen specimen and the liquefaction of the specimen. Okay, so far what we talked about is basically we've got a real acidic environment, okay? We got a lot of acid involved. So we have to have something to neutralize that, all right? Because the vagina has a lot of acidity to it when we're talking about intercourse. The prostate gland, there's a lot of acidic fluid that's coming from the prostate gland. So the semen has to have something that contributes something that has alkalinity to it that's going to neutralize this. This is where the bulbourethral gland comes in on the very next paragraph. It says the bulbourethral gland is located below the prostate. It contributes about 5% of the fluid volume in the form of a thick alkaline mucus that will help neutralize acidity from the prostate and the vagina. Then it goes on to say it's important for semen to be alkaline to neutralize the vaginal acidity present as a result of normal bacterial vagina flora, and without this neutralization, sperm motility would be diminished. So again, let's look back at that picture on page 204, and I'm going to show you guys, I know I just went over a lot of information with you, but that table at the bottom of page 205 is a very good summary. That's what I would study for the test.
Summary 10-1.
It mentions each one of these structures at the bottom of page 205. Summary 10 tac 1 and table 10 tac 1 at the top of page 204. Because you're gonna have a test question that's basically gonna say summarize all this, and it's even gonna say you need to tell what percentage is involved in each part of the fluid. Like for example, if you look at the top of page 204, it says sperm make up 5% of the fluid. Everybody with me? The seminal fluid, that's a large portion. 60 to 70% of the fluid is seminal fluid. The prostate gland contributes 20 to 30%, and then the bulbourethral glands is just 5%.
Question.
Yes.
Once again, the contribution in bulbourethral was the buffer.
Yes, it's an alkaline fluid. It's a real thick alkaline fluid. It's 5%. Okay, so let's look back at that picture on page 204. So we at least, I'm not expecting you to be an expert, but we need to at least have a basic understanding. I want you to look at the top picture on page 204. You see where the testes is, right? That's where the sperm are going to originate from. You see the epididymis is where the sperm are going to mature at. They're going to develop flagella right there, okay? Now you've got the ejaculatory ducts, or what's called the vas deferens, okay? For any of you that want to know, that's what a male has messed with or tied or burnt or cauterized when they have a vasectomy, all right? And that's how come, again, you guys may already know this, but I want you to kind of make sense of this. If a male has a vasectomy, it's not that they don't produce semen, okay, or the fluid. They produce all the components of the fluid except for the sperm, okay? And it's not that the sperm are dead. There are no sperm. That makes sense? That's how come when they go get a post-vasectomy analysis, the fluid is still there, but they have to really, really check and make sure there is no sperm in that fluid, because they cauterize that little tubule, that vas deferens, and so there are no sperms to go through that duct. But all of those different components we've talked about, like the prostate, the bulbourethral gland, the seminal vessel, all of that still contributes to fluid, so it's missing everything but the sperm, okay? That's one reason everyone is supposed to, after they have a vasectomy, the tech is making sure that there are no sperm in that specimen, okay? All right, so make sure that you can locate the seminal vesicle, and then you see the big prostate. That's what all males usually have trouble with when they age. Your prostate gland starts to swell up a little bit as you get older, and because of that, it pushes in and it causes it harder to void urine a lot of times. So it's just a natural aging process in males a lot of times, that they have to go to the restroom more frequently, and it's a little bit harder. You lose a little bit of your urine flow because you can see where that prostate is located, okay? If it swells up, it starts putting pressure to where you can't void urine as easy as you can when you're younger. All right, everybody good on that?
Mm-hmm.
Okay, another objective that we need to talk about specimen collection. On the left-hand column on page 205, halfway down, go down about 3 lines, and it says most of the sperm are contained in the first portion of the ejaculate. So if you look at the very last sentence in that paragraph, it says patients should receive detailed instructions for specimen collection. Next sentence says specimens are collected following a period of sexual abstinence of at least 2 days to not more than 7 days. Now make sure you read this chapter. There's a lot of stuff that I'm having to skip over for time constraints, okay? But that'll make this make a little bit more sense. Always read those technical tip boxes. I think those help out as well. Okay, semen analysis, page 205, right-hand column, go a little over halfway down. It says semen analysis for fertility evaluation consists of both macroscopic and microscopic examination. The parameters that we're going to talk about today are going to be what should the appearance look like, what should the volume be. We're going to talk about viscosity of the specimen that can tell us a lot. There's a normal pH to the fluid. We're going to have to determine a sperm concentration and count. So we're going to have to talk about the hemocytometer again, because a lot of sperm counts are done on a hemocytometer. And believe it or not, they're gonna count different squares, so we're gonna have to figure out how to do those counts, right? Also morphology. You can actually do a right stain on semen and you can look at the sperm and you can tell abnormal sperm. There's pictures in your book that's going to show you sperm that have double tails, that have double heads. There's some that will have a bent neck, okay? It's not uncommon to see an abnormal sperm or two, but you don't want over half of them to be that way, right? Because it only takes one for conception. But we're going to show you different pictures of what sperm can look like. You can have coiled tails and all kinds of different stuff, all right? And you can see all that by actually staining the specimen. And the stain actually is the same stain that we do a right stain on, where you guys did your differentials. Okay, so the appearance. Last little section on page 205, it says normal semen, and I think that this is an objective. Normal semen has a gray-white color to it. It will appear translucent, and it has a characteristic musty odor. And I think another objective is going to state, okay, well, if that's what a normal specimen looks like, we need to know what abnormal specimens look like, right? Because that's probably why the physician's testing, okay? So, skip a line that says increased white turbidity. If you have a specimen that has increased white turbidity, this can indicate the presence of white blood cells and infections within the reproductive tract. Skip on down about 5 or 6 lines and it says varying amounts of red coloration are associated with the presence of red blood cells, and of course this would be abnormal. And it goes on to say, well, what about a specimen that is yellow? It says yellow coloration may be caused by urine contamination. Also, it tends to be more of a yellow color after prolonged abstinence. And it also tells you there, remember that urine is toxic to sperm. So the sperm, if it has urine in it, it's going to be immotile. Okay, so that would be a reason to reject a specimen. Okay, if it's contaminated with urine, the urine are obviously going to be non-motile, and you don't want to turn that out as that person's going to have trouble with conception. All it is their sperm specimen. Or I'm sorry, urine. Medications can also cause the urine to be yellow in color. I think I skipped over that. Okay, liquefaction. It says a fresh semen specimen is clotted and should liquefy within 30 to 60 minutes after collection. So it is very important that if they collect this specimen at home, all right, they have got to write down what time they collected it at. Because if that specimen does not liquefy within an hour, that can indicate infertility, all right? So if they write down the wrong time or don't write down the time, that can be a major problem. Now, you may be working at a fertility clinic where they have special rooms and all that stuff where you can keep up with it. But if they bring that specimen from home, it is very important they write down the right time, okay? Now, again, it's important to pay attention to this. You know, you may not ever have to do this, but you may go to work somewhere where this is what they specialize in, and this is all you do. I actually think it would be a good start to a career to learn how to do this stuff, because not many people are going to know how to do this, which basically means you're going to make more money. Okay? Okay, under volume, the next section on page 206, normal semen volume ranges between 2 and 5 milliliters. So basically, it's going to go ahead and tell you that a lot of times you're going to want to pour the specimen over, and they have a real small graduated cylinder where you can determine what the specific amount is. It does go on to say increased volume may be seen during periods of extended abstinence. Decreased volume is usually what you have to worry about. It says this is frequently associated with infertility. It can indicate improper functioning of one of the semen-producing organs, like the prostate, like the vulvarictal glands, or the seminal vesicle. It says primarily the seminal vesicles is what will be causing this. Incomplete specimen collection must also be considered. All right, under the viscosity section. Specimen viscosity refers to the consistency of the fluid, and it may be related to specimen liquefaction. Skip a sentence that says the normal semen specimen should be easily drawn into a pipette and form small discrete droplets that do not appear clumped or stringy when falling by gravity from the pipette. You'll skip a line that says ratings of 0 to 4 can be assigned to the viscosity report. Now, I'm just going to tell you, a normal specimen, if you start trying to test that before the hour is up, in other words, you have to let this liquefy before you can do this test, all right? So you don't want to test it for its viscosity before that 1 hour time limit. Or, of course, it's going to have too high of a viscosity, all right? So you want it to liquefy. Okay, under the pH section, go down to the 4th line under pH, and it says the normal pH of semen is alkaline and should range between 7.2 and 8. It says if you have an increased pH, this can indicate infection within the reproductive tract. A decreased pH may be associated with increased prostate fluid, duct obstruction, or poorly developed seminal vesicles. Now, in case I forget, I just want to tell you, now again, I've never did this, but this book and all other books are going to tell you, you can actually measure the pH of the semen with your urine dipstick. Because there's a pH pad on the urine dipstick, right? Remember? You can actually use that to determine the pH. You can also use the pad on the dipstick for leukocytes in case there's an infection, all right? That will show you if there's a lot of white blood cells in the specimen. Okay, under sperm concentration and sperm count. I know you guys don't like seeing it, but you see the hemocytometer grid over there, right? Okay, so we got to talk about if we're going to do a count on a semen specimen, which squares do we count? Is there a dilution factor and all that kind of stuff? The first thing that I'm going to tell you is when you go home and study, you need to read this, because I'm just going to make it short and sweet to where it makes sense, or hopefully it does. You can't do a count on sperm that are moving. That makes sense to everybody? You don't have to study that. If those sperm are moving and they have a lot of motility, how in the world are you going to count them on a hemocytometer, right? Because they're going to be up going crazy. So you do have to put something in the solution that is basically going to kill the sperm, okay? So they're still, so you can count them. So you're going to load a hemocytometer. The book is going to tell you normally, for most reasons, they want you to do a 1 to 20 dilution, okay? Because think about how many sperm there are in a specimen, okay? You dilute it so there won't be as many, but we got to make up for it when we calculate the result, okay? So usually you use a 1 to 20 dilution, right? I'm really not worried about what you dilute it with, but the book is going to tell you the substance that you use to make a dilution. I am worried about the fact that you guys know how to make a 1 to 20 dilution, okay? Do you remember how to do that? How would we make a 1 to 20 dilution? Just anybody tell me, just make it as simple as you want to make it.
1 part semen, 19 parts solution, or
Great.
whatever.
Great, that's okay. So as long as you do that, you make that dilution. Now here's the grid, and this is in your book. So we're gonna call this, we're always gonna do for right now a 1 to 20 dilution on the specimen. So here's my grid. Now, you remember when we used to do the platelet count, and we would count all 25 squares in the center square? Even when we did the red count, if you'll remember, this is the squares that we counted. We counted the 4 corners and the one in the middle, okay? These are the squares we count for the semen analysis, all right? So for right now, I just want every time when you think about counting a specimen for a sperm count or a sperm concentration, we're going to do a 1 to 20 dilution. We're going to get our hemocytometer grid in real good focus. We're going to center in on the very middle square, and we're only going to count the corners and the one in the middle. All right? You're going to count this side, and then you're going to go to the other side, and you're going to count it, and you're just going to take the average. You don't add them together. So let's just say we counted 30 on one side and 30 on the other side. So the number that we're gonna work with is 30. If you do it this way every time, you use a 1 to 20 dilution and you count these squares and you take the average, whatever number you get, you add 6 zeros to it, okay? So for example, if I counted 60, all right? If I counted 60, my answer is gonna be 60 million. Okay? This answer, one of the things that's gonna distinguish this from all other counts, this answer is in milliliters, not microliters. Up until this point, every count we've ever done has been in microliters. This is done in milliliters. So even if you don't understand it, and you don't understand the math, as long as you do a 1 to 20 dilution, as long as you count these squares only, take the average, however many sperm you count, you add 6 zeros. Right? And it tells you that in the book. It's just, I don't like to explain it exactly the way they are. The one thing that will get you slipped up on this is if you say this answer is in microliters. Okay? This is not. This is per milliliter. Because for whatever reason, for sperm concentrations, they want it in milliliters, not per microliter. So if we said that we had a count of 60 million per milliliter of semen, okay? In other words, there's 60 million sperm in every milliliter of that semen specimen. That's the concentration, okay? Now listen to me here, guys, because there's a test question that'll eat you up if you don't get this, all right? That's the concentration of it. But if I asked you for the total sperm count, in other words, how much sperm is in the total semen specimen, all right? Somebody tell me, how do you think you'd figure that out? Great, we gotta know how much specimen there is, right? So if we know that there's 60 million sperm per milliliter, let's say that my specimen, or whoever's specimen, is 2 milliliters. You just multiply that number times 2, right? That would be the total sperm count per ejaculate, or per total semen specimen. Everybody with me on that?
Multiply by 2.
So this is the answer as in the concentration. But if I asked you for the total sperm count, you would have to multiply this number by how much volume of semen that there was.
We don't have to worry about the dilution factor, like multiplying it back by the dilution factor since it was diluted 1 to 20.
No, but we could. I can get the same number by showing you all that.
It's incorporated, all that rigmarole is incorporated into the 6 zeros.
Yep.
Okay, cool.
But we're going to do one here in a minute where we're going to do all that. Okay, because what I'm afraid of is on your registry, and I know you guys have heard me say this before. I don't think they'll give it to you this way. I think they're going to want to make sure that you understand the hemocytometer and understand the math part of it, because there's going to be a question in the back of your book that, or actually I think it's in your reading, to where they say, look, you don't count these squares, you just count 2 large white cell squares. Now we got to plug in the dilution. We got to figure out how many squares we counted out of 10. You remember that saying I always tell you, what would you have counted? Remember if you had counted 10 large squares?
Ugh.
All right, so let's do that problem. So does everybody feel like, and I'll give you one on the test. I'll give you one like this. I'll give you an easy one, okay? To where even if you don't understand the math, you can say, look, Mr. Britt said the dilution 1 to 20 told me to count these very squares right here. What's the sperm concentration? It's just however many you counted, you add 6 zeros, okay? That's simple. But if you truly understand a hemocytometer and understand the count process, you should be able to do this next problem. Now, I'm not going to do it the way the book does. I'm just going to tell you that right now. You're welcome to do it the way the book does, and you'll get full credit for it on the exam. Let's look on page 208 under the examples. I'm up at the top of page 208. Look at question number 2. It says in a 1 to 20 dilution, 600 sperm are counted in 2 white blood cell counting squares. Calculate the sperm concentration per milliliter and the total sperm count in a specimen with a volume of 2 mils. Okay, so this one is done different, right? Does that say micro or mils? It says
Micro.
milliliters. Okay, it needs to be corrected. It should be 2 milliliters. I've got that written in my book, correction. It should say 2 milliliters. That's the total sperm volume. There's not going to be 2 microliters, right? That'd be too small. Okay, so let's talk about this. It says we're going to do a 1 to 20 dilution.
That needs to be milliliters.
This is going to be our dilution. And it says that they counted 600 sperm are counted in 2 white blood cell counting squares. Okay, so they're going to count this. Let's just say we count this square and this square equals, how many did they say they counted? 600?
Yep.
Is that right?
Yep.
Okay. Now, the first answer we get is going to be per microliter, because now we're going back to blood cell counts, okay? So, Kennedy, work with me on this one. We counted 2 large squares, right? So how many do we have to count to actually get a good result? How many total squares should we count altogether? Okay, that's all right.
10.
Anybody remember when we
10.
had 10?
It's always 10.
Always 10.
Yes.
Good. We want to get to 10 large squares, right?
Always.
Because 10 large squares is a microliter, all right?
Multiply by 5.
So if they counted 602 large squares, how many would we have counted if we would have counted 10 large squares? What would we multiply that number by?
5.
5, okay, so what's 600 times 5? 3,000. So if we would have took the time to count 10 large squares, like if I was working in the lab, I would count this complete side, and then I would go to the other side and count 1 more large square, okay? Or if you want to count 5 on this side and 5 on the other side, okay? Just get to 10 by whatever way you want to do it, okay? So if 600 is equal to 2 large squares, we just determined that 3,000 would be to 10 large squares. So we'll mark this out. 3,000 equals 10 large squares. All right.
Okay.
Right here, since 3,000 would be how many we would have counted in 10 large squares, the only thing left to do, we got to remember what the dilution is, right? The dilution's a 1 to 20, right? That means they diluted it, 1 to 20. So we got to take this number and multiply it times 20. What's that going to be?
Yep.
60,000. Everybody with me on that? When we get to this point right here, you have done the count, but remember, sperm concentrations are done per milliliter. This is the answer of how many sperm are in each microliter, all right? So now to get to milliliters, what do we have to multiply this number by?
100.
You got to add 3 zeros.
Yep.
Okay, right? Got to add 3 zeros.
Wait,
so we'll take 60,000 and we'll add 3 zeros. So in my mind, the sperm concentration for this particular problem is going to be 60 million per milliliter. Is that what it says in the book?
Yes.
Okay. Now if we want to know the total sperm count, how much volume was in this specimen? 2 mils. So the total sperm count should be 120 million per ejaculate. Okay? So when you take the test, if you're reading the multiple choice question and it says you count the 5 little squares in the red blood cell counting area and it says your dilution is 1 to 20, all you got to do is take the number that they come up with and add 6 zeros. That'll be your answer per milliliter. But if they throw this at you and say they counted 2 large white cell areas, now you have to be able to do the math in your head to figure out a hemocitometer count. You know, for a long time now, I've been telling you guys, Look, I did not want to learn this way when I was a student. I fought it every step of the way. I'm like, no, I want to know that easy way where you just add 6 zeros. But if you can do this math, the reason I say it's so important, if you will always figure out how many would you count in 10 large squares, you can do a sperm count, you can do a red blood cell count, you can do a white blood cell count. It doesn't matter because 10 squares is 10 squares. It doesn't matter what you're counting, okay? Like, I'm not gonna do it because I know that this stuff is hard when you're first learning it. I could take that first problem where we just added 6 zeros, you know, where they counted the red blood cell squares. You could do that in your head and go, okay, they counted 5 little squares in the center square. You would multiply that number times 5, right, to take up the whole square, because there's 25 little squares in that middle square. Then you take that number and multiply it times 10 to make up for 10 large squares, you will get the same answer. Okay.
But still, you're taking the average on the beginning. From what you're counting.
Now, when you study for your test next Friday, and the reason a lot of you need to make sure that you do this extra credit I'm telling you about, 10 extra points added to a test score, is because now you're going to have to be able to distinguish a white blood cell count on spinal fluid versus a sperm count on semen analysis. All right? So you got a lot of stuff to you got a lot of math to look at. And I'll be happy to help you with it one-on-one any free time we have next week if you're having trouble with this. And there's usually always a review question in the back of the book that'll ask you a question so you can see if you're doing it right.
Yes, sir.
Okay, sperm motility on page 208, left-hand column, down toward the bottom. So we're evaluating sperm, even though we can do a sperm count now, because see, there's normal ranges for sperm, okay? You can have a low count, which basically is going to show you infertility. Normally you don't have high counts. Normally they're worried about low sperm counts, males. But the sperm that are there, they have to be moving, right? They have to be motile. Remember what we talked about when we were talking about the composition of the fluid? What's that one thing that is a big help in making them motile? They metabolize that sugar.
Fructose.
Fructose. That comes from the seminal vesicle, okay? All right, so sperm motility on page 208, it says the presence of sperm capable of forward progressive movement is critical for fertility. Because once presented to the cervix, the sperm must propel themselves through the cervical mucosa to the uterus, the fallopian tubes, and then basically to the egg. So when you read over this section, it'll show you how to grade forward progressive movement. There is a section I think that is important in the right-hand column on page 208, go down about 10 or 11 lines. It says grading can be done using a scale of 0 to 4, with a 4 indicating rapid straight line movement and a 0 indicating no movement. Right? And it refers you to table 10-3 at the bottom. It says a minimum motility of 50% with a rating of 2 after 1 hour is considered normal. And you can look at table 10-3 and it'll say, for example, a grade of 4 is rapid straight line motility, a 3 would be slower speed, some lateral movement, and then a 2 would be slow forward progression, noticeable lateral movement, and a 1 would be no forward movement. Okay, so how do we do this? I mean, how do you literally, like, if you've got little lanes and you're trying to figure out what percentage of the sperm are motile, and even if they are motile, are they moving the right way? Okay, now guys, don't look in the book right now, just listen to me, okay? Think about this. We've got to figure out of that semen specimen, how do we just look at the sperm and figure out, okay, a large percentage of these are moving in a straight line, okay? What you do is it's just like doing a urine sediment. Just take a drop of the fluid, put it on a microscope slide, use a 22 by 22 cover slip. I think that's what the book will tell you. And then you just look at it. You look at like say 20 fields, okay? And you try to figure out, and this is why it's considered a high complexity test. There's a lot of interpretation involved in this, okay? And you're gonna look at it and you're gonna try to figure out of the sperm that you're seeing, what percentage has basically forward progressive movement. I mean, obviously, if you look under the cover slip, now you didn't add any type of chemicals to kill the sperm now, right? We're looking at the real deal, all right? You're looking to see, is there a high percentage of those sperm that are moving forward, okay, and at a constant rate? Like if they're all moving this way, right, to the side, that's really not gonna work. That makes sense?
Right.
They've got to have rapid progressive movement. So you can look at these under a cover slip to find that out. Now again, I've never done one, but I can just tell you that what I would do is I would look at that and I would look at several fields and I would make an estimate. Like, look, it looks to me like about 80% of them look like they're moving just fine, forward progressive movement. Okay. I am going to go on to talk about here in a little bit. Thank goodness they have a thing called a CASA, Computer Automated Semen Analysis Machine now that does this stuff for you, and it's not so manual. So if you were to go to work at a fertility clinic, I would just about bet you they have one of these analyzers that do a lot of this. Okay. All right, so let's look at sperm morphology on page 209. Sperm morphology, go down to the second full paragraph, well, go down to the first full paragraph under sperm morphology. It says sperm morphology is evaluated with respect to the structure of the head, the neck piece, the mid piece, and the tail of the spermatozoa. It says abnormalities in head morphology are associated with poor ovum penetration, whereas the neck piece, the mid piece, and the tail usually have to do, or they affect the motility. It goes on to say the normal sperm has an oval-shaped head approximately 5 micrometers long and 3 micrometers wide, and a long flagellar tail approximately 45 micrometers long. Says critical to ovum penetration is the enzyme containing acrosomal cap. I always call it a toboggan, all right? Think of the sperm wearing a little toboggan, all right? That little toboggan has special enzymes that help it penetrate, right? You can see it on that picture down there below. It's kind of purple color. Says this will be located at the tip of the head. So you've got what a normal sperm looks like at the bottom of that page. And you can see the acrosomal cap. You can see the cell membrane, the nucleus. See the mitochondria? That's what gives, basically that's like the energy center of the sperm, all right? And that's what's going to give the tail energy, is that mitochondria section. As long as that's working, that tail is going to be able to move. Now, one other thing that I want you to make sure and have highlighted in your book is when we're evaluating sperm morphology, we actually have to stain it, okay, to look at it, to be able to see a lot of the different structures. So I'm going to direct on page 209, go to the right-hand column and go about halfway down in that big paragraph right there. And it says staining can be performed using a right stain. Everybody see that word rights, comma, and it says GIMSA. GIMSA is a stain that's like, it's another name for a right stain. Okay. And then it also mentions shore. I've never heard of that, and that will not be included, but and also it's got a papiniculo stain. Those are 3 different stains that can be used to observe sperm morphology. So if you see a test question next week that says, name any stain that can be used to observe sperm morphology, if you tell me a right stain, a GIMSA stain, or a papiniculo stain, you'll get credit. You don't have to know all 3 of them, but know at least one. Okay? And I'll say that again during the review. So if you missed that question, it's on you. Right? let's look at some of these pictures just to show you things that can be wrong when you're observing sperm morphology. Let's look at figure 10 tac 4. What's wrong with that sperm right in the middle?
Double head.
Yep, it's got two heads to it, right? So that's an abnormal sperm. That does not mean that you are not going to be able to conceive. That just means one out of the many, many sperm that you're looking at happens to have a double head.
Okay.
What about the one in the picture below it? What's wrong with that one?
That's strange.
They call that an amorphous head.
Its acrosome is messed up.
Yes, that will definitely affect the toboggan. I'm not taught that. I will on the test, but I won't when I'm talking. All right. What about the picture on page 210? There's a couple of things wrong in that picture.
Double tail.
I see one with a double tail.
One that has like super skinny and one bent.
One of them has that bent neck. You see that one over there at about, I guess, whatever you call it, 10 o'clock, or you see that one where you got the head and then the tail, like it's not kind of behind it. It looks like it's broken.
It's like an L shape or whatever. Okay.
Hey, look at the picture on page 211. I want you to look at that big, look at that cell in the middle that looks like a lymphocyte. You guys see that?
Mm-hmm.
That is an immature sperm. That's called a spermatid. Okay? Sperm develop just like white blood cells, all right? I mean, before they mature, they don't look like a normal sperm with a head and a long tail or a long flagella. So that's a spermatid. And when you read this, and I know you're all going to read it, there's going to be a section in the book that says, look, if you see a lot of those, you have to be able to distinguish those between white blood cells. What if there's lots of white blood cells in the fluid, then this person has infection, right? So you can see those white blood cells when you do the stain, but you have to figure out, are they white blood cells or are they spermatids? Because they kind of look alike, right? Like, are you truly looking at a lymphocyte white blood cell, or are you looking at a really, really young spermatozoa, a spermatid? Yeah.
Oh, so in 10.8, the spermatid is the biggest dark purple thing?
In 10.8, yep. That one that looks kind of like a lymphocyte. That's not a lymphocyte. That's
So as the spermatid matures, it increases in size.
yes.
Okay, cool.
Where is it? Let's go back. Let's have a little quiz. Where is it that the sperm mature at out of those organs that I told you about? Epididymis. Remember that little layer that's sitting right up on the testes.
Above the testes. Okay.
But see, you can have a problem. Let's just say that the sperm are actually going through the tract before they come to maturation. Okay, then you're possible, or you could possibly see a spermatid in a semen specimen. You don't want to see too many, right? I mean, again, I've never did one, but I'm sure that you may see a few spermatids, but you're not going to actually, you don't want to see a large number of them, just like you don't want to see a large number of white blood cells in that semen specimen, right? Because that can indicate infection. Again, you have to kind of keep remembering in the back of your head, there is a reason you're looking at that specimen, right? Because they don't just have you do this if you're going to be normal. So you have to be looking for stuff like this, because obviously they think something's wrong or they wouldn't be submitting the specimen. The only exception to that is a vasectomy, okay? Because basically then you're not looking for any sperm.
Right.
Okay, sperm vitality on page 211, left-hand column. Says decreased sperm vitality may be suspected when a specimen has a normal sperm concentration with marked decreased motility. There is a stain. That basically, guys, that means that the majority of your sperm are dead. Okay, like you've got lots of sperm in the semen specimen, but you need to figure out, are they all alive or are they dead? There is a stain that you can use if you'll drop down about 2 or 3 lines. It's called an eosin nigrosin stain. It says vitality is evaluated by mixing the specimen with an eosin nigrosin stain, preparing a smear, and counting the number of dead cells per 100 sperm. You can use bright field or phase contrast microscopy. Living cells are not infiltrated by the dye and they will remain blue-white, whereas dead cells will stain red against the purple background, like that picture right beside it. You guys see those pink cells in there?
Mhm.
Those are indicating dead sperm. And it goes on to say a little further down in that paragraph, it says normal vitality requires 30% or more living cells and should correspond to the previously evaluated motility.
30%? Or,
You're right, 50.
I'm good, right?
50, yes, I can't
No,
see.
it's, I'm
Correct.
not
It
correct.
says 50. I'm good that you can see that it says percent. Alright,
Just didn't know my book had an error.
alright guys, you can bear with me another 10, can we go another 10 minutes?
Oh, yeah.
I'll have this knocked out. All right.
For sure.
Anti-sperm antibodies on page 212. When you read over this section, basically you just need to understand one thing that can cause infertility. It can be due to the male or the female, okay? Now I'm gonna let you read this on your own, but what I get out of it is, if a male has a procedure done, like the testes, okay, are supposed to be separated from the male's immune system, okay? The testes are dropped down into a sac, they're really kind of outside the body, okay? But if a guy goes in and has, say, a vasectomy or a vasectomy reversal, where they're doing incisions and stuff down in that area, it can actually cause the testes to actually be in, it breaks the barrier, okay? If that barrier is ever broken, the immune system sometimes does not like what's going down in the testicle area, okay? And a male can actually develop autoantibodies to their own sperm.
Wow.
So that can cause problems with fertility. It has nothing to do with the female. The male actually has an autoantibody, okay, that is attacking their own sperm. And when that autoantibody coats that sperm, basically it's going to become ineffective, okay? Or when it coats that sperm, then when the male and the female get together, now the female's immune system sees an antibody coating a sperm and they develop an antibody to that, okay? So they can do tests to determine whether it's a female has an antibody to the male's sperm or whether the male has an autoantibody to their own sperm. That's what they do at fertility clinics when they find out two people when they can't conceive, all right? Now that reading is going to tell you about a MAR test and immunobead test that they can do to determine where the antibody is attaching to the sperm and all of that. But read over that section on your own. Yeah, I've never done that test, okay? But I'm just telling you that fertility, a lot of times, you have trouble with fertility due to antibodies. It can be in the male or the female, so make sure that you read over that. Microbial testing, you'll go down about halfway on the right-hand column on page 212. It says, on certain occasions, the lab may be called on to determine whether semen is actually present in a specimen. A primary example is in cases of alleged rape. Okay, this is going to usually be due to a wet prep. If somebody comes to the ER and they're doing a rape kit on them. Go down about 6 or 7 lines further in that paragraph. It says seminal fluid contains a high concentration of prostatic acid phosphatase. That's usually going to be abbreviated PAP. And it says, so detecting this enzyme can aid in determining the presence of semen in a specimen. They always ask this in textbooks, and I don't know why, but if you have a fluid down in the lab, and let's say you walk up to it, it doesn't have a label on it or anything, and you don't know whether it's amniotic fluid, you don't know whether it's spinal fluid, you don't know whether it's semen, you can do a test on that fluid. If you test it for prostatic acid phosphatase, that enzyme is generally only found in large amounts in semen specimens. So that would be a test that you could do on that fluid to determine if it was semen. Will you ever have to do that? I doubt it, okay? Because most places, if you have a specimen that's unlabeled, if they can't figure out right away what the problem is there, a lot of times they will pitch that specimen, okay? Because you just don't want to make sure you're not trying to get patients mixed up on certain specimens. I mean, let's think about it. If we work at a fertility clinic, all you're gonna be working with is semen specimens, right? So if you find one that's unlabeled, somebody's probably in a little bit of trouble for that. Okay, post-vasectomy analysis. I really think that most of you, if you go to work in hospitals, this will probably be the only test you do on semen specimens, okay? Unless you go to work at an andrology clinic somewhere. I'm on post-vasectomy semen analysis on page 213, left-hand column. It says post-vasectomy semen analysis is a much less involved procedure when compared with infertility analysis because the only concern is the presence or absence of sperm. It goes on to say specimens are routinely tested at monthly intervals beginning at 2 months post-vasectomy and continuing until 2 consecutive monthly specimens show no spermatozoa. So basically you look at a wet prep and see if there's any sperm. If you don't see any sperm, you take the specimen, you spin it down, okay, you concentrate it, you spin it down in a centrifuge, and then you get the sediment, if there is any sediment, and then you look to see if there's any sperm in that. In other words, a post-vasectomy, you should see no sperm.
Just to refresh, again, what is a wet prep?
A wet prep is it's like vaginal fluid. You take a drop of vaginal fluid and you just put it on a microscope slide and put a cover slip and look at it.
So some fluid, it doesn't have to be vaginal.
Well, for the most part, it's vaginal. Whenever you think of wet prep, it's a vaginal specimen. You probably won't. A lot of times they do that back in microbiology, but anytime I hear the word wet prep, I'm thinking, are they looking for like a yeast infection in the vaginal fluid? Or are they looking for, unfortunately, in cases of, like I said, yeast, Candida albicans, anything like that? But a wet prep is usually done back in microbiology.
You just mentioned wet prep in this context.
Mm-hmm.
Say that again?
Yeah, you're gonna look at a wet prep for the presence of sperm, okay? Or, okay, sometimes they'll associate a wet prep with just adding a drop of semen on a microscope slide. That would also be considered a wet prep. Just the semen itself. Because remember, okay, if somebody's had a vasectomy, they're still going to produce semen, okay? It's just there's no sperm in that semen. It has all the other components, all right? And so what they do, just to make sure and that you can't go back, you know, if somebody has a vasectomy, they're not wanting to get pregnant, right? Okay, and so they want to make sure of that. So after they have that procedure, they make them come back, like the people that do actually come back. They make them come back, and then they make sure that the procedure was effective. And so they're going to have you produce a specimen, usually 2 months out, and sometimes 3 months. Thank you. Under semen analysis and quality control, I think this is important. It says traditionally routine semen analysis has been subject to very little quality control. Going over to the next column, it says the analysis is rated as high complexity test under CLIA, and testing personnel standards must be observed. They do, you know, back when I worked, they did not have QC material for this, but they have developed QC material for it now. So you remember when I was telling you periodically, like once every 3 months, you're gonna get those styrofoam CAP surveys? They have CAP surveys now for people that do tests on semen. All right? Okay, let's look at some of these review questions. I don't have these answers, but I'll read over them real quick. Let's look at page 214. Let's go over some of these. It's going to take me just a minute because I need to read them first to make sure that we at least went over some of them. Okay, number 1, maturation of sperms takes place where?
C.
Epididymis, right? Number 2, enzymes for coag and liquefaction of a semen specimen are produced by which? The prostate gland. The prostate gland produces enzymes that basically allow for coagulation and liquefaction of the specimen. Number 6, liquefaction of a semen specimen should take place how long?
Within an hour?
1 hour, good. When you read, you're reading, I didn't cover it today, but it's going to tell you, remember I told you, of course, these specimens are usually collected by masturbation.
Mm-hmm.
Well, some people, they don't want to do that for various reasons, and so they collect it in a condom, okay? And if you collect it in a condom, you got to remember most condoms have germicidal things in it and stuff, so you can't collect it in a condom that has any of that. I'm assuming your fertility clinic would provide the appropriate condoms,
okay.
but you can't just use any condom to collect that.
So that addressed question 7?
Yes. All right, I think we got a problem here. Hold on. Number 11. Let me think about this real quick. We may do this one. It says, given the following information, calculate the sperm concentration dilution. The dilution is 1 to 20. The sperm counted in 5 red blood cell squares on each side of the hemocytometer is 80 and 86. The volume is 3 mils.
So you take the average of 80 and 86, correct?
Okay, so let's do this problem. Number 11. I'm on number 11 on page 214. I think we can do this in our heads. I don't even think we got to use a calculator. All right? Let's try it. We'll read it again. Read it slow. Given the following information, calculate the sperm concentration. So when I say sperm concentration, they are wanting to know how many sperm are in every 1 milliliter volume of that specimen. Remember, sperm concentration, it's not per microliter, it's per milliliter. So that's going to be the answer to this question, is how many sperm per milliliter. Now, if I asked you for the total sperm count, it's how many sperm per ejaculate, all right? All right, so it says we're using a 1 to 20 dilution. Remember, that goes with what I said. That's totally standard. Yep. It says sperm counted in 5 red blood cell counting squares. On one side, they counted 80, and on the other side, they counted 86.
So 83 is the average.
Everybody agree with that? 83
yes.
is the average? All right, so we already know the answer. If 83 is the average, what do we do?
Add 6 zeros.
Add 6 zeros. So what's the answer?
83 million.
83 million. Is that one of the answers? Okay. Well, hopefully that's the answer. 83 million per milliliter. See, you can automatically, and guys, I told you, there's always a way, always give yourself the best advantage you can, but before you even do this, you can exclude D, right? Because D is per microliter. Sperm concentrations will never be per microliter. Number 13. Number 13, the primary reason to dilute a semen specimen before performing a sperm concentration is to. Now, I did not directly go over this with you guys, but use common sense here and tell me what the answer's got to be. B? You say B? B, not B.
It's not B. It's to immobilize the sperm.
To immobilize the sperm. The stuff in that diluent that you're using basically kills the sperm. You can't do a sperm count if they're moving. Just trust me, if you look in there, you're going to go crazy. Okay? You can't do that. They've got to be immobile in order to count them. Number 17, I'm just glancing at some of these. The purpose of the acrosomal cap is to, okay, good, penetrate the ovum. Number 18, the sperm part containing a mitochondrial sheath is the B? C, the midpiece. Number 19, all the following are associated with sperm motility except A. Good, the head. Number 20, the morphologic shape of a normal sperm head is C. Oval. C. Number Number 30, on page 216, following a negative post-vasectomy wet prep, the specimen should be centrifuged and re-examined.
Yes.
You are just double and triple checking, making sure there are no sperm in that specimen. Okay, now we've been in here, I went way over. Let's take a 15-minute break. We'll come back and I'll give a review